Good morning, everybody. I hope all of you are staying home and staying safe. Our today's topic is microsporogenesis. Before we go on to the process, let us recap the parts of a flower. So, here you have the longitudinal section of an angiosperm flower, the outermost whorl comprising of the sepals, and inner to it you have the petals. These are actually the non-essential parts of the flower and not directly involved in the reproduction. They are serving other purposes for the flower. Now let us concentrate on the sexual parts of the flower. The stamen, that is made up of the anther and the filament, and we have the carpel, which is made up of the style, stigma and the ovary. So in this chapter we are going to learn about the microsporogenesis that happens inside the anther. So the male reproductive unit, or otherwise known as the stamen, as we saw in the diagram, it has the long slender stalk known as the filament, and then the anther. You might have experienced a yellow powdery substance sticking onto your hand when you handle the flower. That is actually the pollen grains that have um, come out, that have got released from the anther lobes. Okay, now let us see the structure of the anther. The anther is a bilobed structure, and in each lobe there will be two microsporangia. So on the whole there will be four microsporangia on the anther, and inside these four microsporangia only the pollen grains are produced, and the four microsporangia get transformed into the pollen sacs. Now let us see the picture or the structure of this cross section of the anther lobe. This we have already seen, the filament and the anther that makes up the male part of the flower. The cross section of it, a cross section of this anther, is what you are going to see in this picture. Cross section of the anther showing the microsporangia. The four microsporangia are able to see 1, 2, 3 and 4. Four microsporangia inside which the pollen grains are produced. These from these pollen sacs, the mature pollen grains will be dehisced out when the wall breaks open. Right. A closer structure of this microsporangium gives us the details of the wall, which is made up of many layers, and the sporogenous tissue that is present inside. From the sporogenous tissue, the cells will be producing the pollen grains. So, what are the walls of this microsporangium? The walls, the layers that are present are the epidermis, endothecium, middle layer, and tapetum. Epidermis, endothecium, and middle layer are mainly concerned with the protection, whereas the innermost layer called the tapetum is concerned with the nourishing of the pollen grains. And therefore, this cells of the tapetum will be very large in size, will be having dense cytoplasm, and so on. Most of the one more question is about this tapetum, you should know. And then this is the close-up structure. You can see the picture here, again lifted from your textbook. This one shows all the four microsporangia, and the wall of the microsporangia is enlarged here to show all the four different layers. Inside you have the sporogenous cells, which will produce the spores, the male spores or the pollen grains. In this picture you are able to see the mature pollen grains escaping out. Here you are able to see the mature pollen grains dehiscing out of the pollen sac, right? And this is again a close-up picture of the wall showing the epidermis, endothecium, middle layers, and then the tapetum. I told you what is the function of the tapetum. It is to give nourishment to the developing spores. Inner you are having the microspore mother cells that will produce the male gametes, right? uh, what type of cell division do you think will occur to produce the male spores? Yes, meiosis. Sexual reproduction here to produce haploid cells. Meiosis is the type of cell division the mother cell undergoes ultimately to produce four haploid meiotic cells, and all these four in the beginning will be held together forming a tetrad, and later on they will dissociate, right? The four microspore mother cells, they are functional, they give rise to the male gamete ultimately. Here you are able to see two different ways in which the tetrads are produced. Here it's a continuous process here, and whereas here you are seeing the cross walls also being produced, and then successively the cell again divides, the second meiotic division ultimately to produce the four cells. This picture shows the beautiful designs on the pollen grains. Actually, these are pollen grains collected from various flowers. So you are able to find a variety of pollen grains in this picture. Right. It's very beautiful, and it's very nice to observe those under the microscope. Coming to the structure of the pollen grain, you can see it is the male gametophyte, and the two layered walls are there. Outer one we call it the exine, and then the inner wall is called intine. Exine is made up of a substance called sporopollenin. Sporopollenin is the toughest substance known, in the sense that it cannot be digested by any enzyme, and destroying it physically also is almost impossible. The intine is made up of cellulose and pectin, whereas exine is made up of sporopollenin. Intine is made up of cellulose and pectin. So this picture gives the cross section of the pollen grain, showing two cells here inside. Now let's come from outermost exine. You can see it is not continuous. In certain places it leaves gaps, that is called the germ pore. And then inner membrane, that's the intine. We have a generative cell. There is one generative cell here which actually goes to form the male gametes, and then here we have the tube nucleus cell, tube nucleus of the vegetative cell. So here you see this picture is taken from your textbook. You should learn the diagrams. You should know to draw the diagrams and label the parts. So here you have the generative cell. This generative cell is spindle shaped, and you can see it is floating in the cytoplasm of the vegetative cell. This is the two-celled stage of the pollen grain. The generative cell further it will undergo mitotic division to produce the two male gametes. So this picture shows the pollen germination. You should be able to recognize all these three diagrams. Now this you can see has started to germinate. The pollen grain has started to germinate here, and you can see the tube nucleus has entered the pollen tube first, germinated pollen tube first, and then the two male gametes are closely following it. In this you are able to see, in this diagram you are able to see the two male gametes which are required for the double fertilization. I am not going to explain the double fertilization to you. You must learnt it already in your previous class. And this picture shows a number of pollen grains that have reached the stigma, and you can see the pollen tubes have grown out, germinated out, and then the male gametes are going into the various ovules. There are many ovules in this flower, and they're going to get fertilized now. In these you can see the male gametes entering into the sac, right? So we pass on to the other uh, information about the pollen grains, where in some countries pollen grains are consumed, taken as nutritional supplements, mainly by sports persons, so that they can get more energy and perform better. Now my question is, if sporopollenin is the toughest substance made up of this sporopollenin is something which cannot be digested by enzymes, then how come the pollen grains are consumed as food? What will happen when it goes into our digestive system? Try to find the answer for that. And then we come to know about the pollen banks. Pollen banks, or like gene banks, we can store the pollen grains in very very cold conditions, like in liquid nitrogen. They can be stored for very very long number of years. Many such seed banks have cropped up, and then we go on to see here the picture of a fossil showing the pollen grain being preserved by nature. Some of these pollen grains are many thousands of years old, but they are preserved intact